Okay.
Sir, 1 minute. Suresh sir, you can read the profile of sir. Yes, sir.
So good evening, one and all. Heartily welcome to today's second day of International FDP and SDP 2020. Today our speaker is Akondi Buchi Raju. He is Assistant Professor Clinical Pharmacy and Pharmacology at IBN Sina National College of Medical Sciences, Saudi Arabia. He is having 15 years of experience in both teaching as well as research. He has a trained pharmacovigilance specialist from the World Health Organization. He also trained in molecular biology techniques and ICSI at University of Kent, Canterbury, UK. So he has various memberships, membered in Secretary and Advisory Committee of FORMD program, IBN Sina National College of Medical Studies. Similarly, he is having life membership in Indian Pharmaceutical Association, Association of Pharmaceutical Teachers in India, and life membership in Indian Pharmacy Graduate Association. So he done his PhD from Andhra University, Visakhapatnam, India. He has guided 18 MPharmacy students and 40 PharmD students. And he also guided for 2 MBBS students. So this is his brief introduction regarding our speaker today, Akondi Buchirazu sir. Sir, 1 minute, sir. I will request our principal, sir, to address you. Sir? Sir?
You already introduced today's speaker.
Yes, sir.
So thank you, Buchraj Garu, for accepting our invitation and accepting to deliver a lecture on evaluation of clinical research. On behalf of organizing committee, I am very much thankful to you, sir.
Thank you, sir. Thank you very much. Thank you, Bapatla College of Pharmacy, for having me here in this wonderful session. So I think this is useful for both the students as well as the faculty teaching the pharmacy. So I'd like to share whatever knowledge I gathered on this subject called evaluation of clinical research. So I just, summarized everything so that we can inculcate the interest among the students so that they can pursue the studies about the clinical research and how it can be done, or what are the various ways and means to evaluate the clinical research. So this is basically a summary of evaluation of clinical research, we can say. So I think, so whatever comments you have, so please post it so that I can answer them at the end of the session. And I think the moderators will allow me to speak about or maybe the answer those questions at the end of the session. This is what I want to have, because I want to answer all of them at the end of the session. So let's I think, with the permission of the moderators and the chair and the principal, sir, I would like to start this topic, right? If you permit, and if everything, everything is working fine, and if YouTube link and everything is fine, I will continue, because I have no idea whether it is live on YouTube or not, right? So if you permit, I will continue.
Please proceed, sir. Going fine.
Yeah, thank you so much, sir. Thank you so much. So as you all know, the clinical research is a very important step to bring the new drugs to the market. And whenever we do a research, whenever a research is done on a new drugs, So they have to follow certain rules and regulations, ethics, and so on. And after completion of the research, of course, they have to publish it as well. Either they publish it in the journal, or they have to submit their reports generated out of this clinical research to the regulatory authorities of that particular country, right? When it comes to United States of America, you call it as FDA, right? So the Food and Drug Administration, which looks into all this, the reports which are generated out of clinical research, and then give the permission. Okay, that is one aspect of the clinical research. Now I'm going to touch upon the another aspect, right, which is why this evaluation is important. Okay. And the lecture learning outcomes of this today's lecture uh, define evidence-based pharmacy approach. At the end of this session, the students will be able to define the evidence-based pharmacy approach, and they also able to describe important parameters for evaluation of the studies, clinical studies. Then list levels of evidence based on study designs. List the levels of evidences. Identify the advantages of controlled experimental designs, right? What are the basic differences between observational and the experimental studies, and what are the advantages of those experimental studies? So this is the basic idea of having this session. This is the basic idea for having this session. And this session will try to cover all these aspects. Okay, so why a search is important, why we have to search for the clinical research articles or a clinical research results, right? Like hope you remember, like one of the 2 days back there is a big news, right? I can show you that as well. So you know that there is a race for vaccine, for COVID-19 vaccine, and Russia 2 days back launched a vaccine by name Sputnik 5, right? Launched by Russia. Hope every one of you know about it. But there are, at the same time, there are reports all over the media as well as in the scientific community about the safety aspect of this particular vaccine, safety and efficacy aspect, right? Hope you all agree with me, right? So the problem with this particular vaccine, everybody say, is where is the evidence for its safety and efficacy, right? So we have to have an evidence to believe any clinical research, basically, right? When somebody say my drug works better, then the next question comes is where is the evidence, right? So what is the evidence for the safety? What is the evidence for the efficacy, right? He must be able to provide that efficacy safety studies, either in the published format or by submitting those reports to the regulatory authority like DCGI or the FDA or whatever regulatory authorities of that particular country, wherever the studies are done. So you need to have an evidence to look into to understand whether that particular drug is safe or efficacious, safe and efficacious or not. So this is the idea why we need an evidence, why we need to search for the evidence, right? Until unless you have an evidence, nobody will believe you, right? So you have to show them an evidence that this is the evidence I have for the safety and efficacy of my study, right? Now let's go back. Right? Let's go back. Why the search is important. A clinician wishes to know if there have been reports. There have been reports of an allergic reaction to insulin and would like to find literature discussing insulin allergies. It's very common that you got a doubt, or maybe a doctor have a doubt in his mind that what is the possibility of getting an allergic reaction with insulin? So is there any, the discussion about the insulin related allergies in the literature? And this doubt is very legitimate, am I right? So once he got this doubt, what he will do? He will go and search for the evidence, search the literature, search the PubMed, search the Science Direct or Google Scholar. For what? To get the information about that. Sometimes a clinician can also approach a pharmacist because, the pharmacist is regarded as source of information about the drugs. Sorry. So apart from medicine management, a pharmacist He is expected to be a great source of advice for clinicians, nurses, dentists, and patients also. Many times patients also reach the pharmacist to ask about a particular evidence, or maybe they ask about what is the evidence for this particular drug. Can you give me a guarantee that when I take this drug, it is safe and efficacious? Now, if you go and ask any community pharmacist or a hospital pharmacist or basically any healthcare practitioner, they will tell you so many questions. The patients pose to them. The patients ask about what is the guarantee that this drug is safe? What is the guarantee that this drug is efficacious? So you have to give them a guarantee. You have to explain them that this drug was well studied, this vaccine was well studied, and there is a proof that this is efficacious, this is safe, there's no problem at all, you can have it, right? So this kind of confidence you have to give to the patient. How you get this confidence to give this to the patient? You get this confidence from the literature which you read, the literature which you look into, and this literature search is very, very important. This literature need to be searched and then need to be evaluated for its authenticity or validity, right? If the literature is true, if the literature is validated, if the literature is evaluated, then you can believe that literature, and that whatever literature you have, whatever the evaluated literature you have in your hand, if that literature says that this drug is very safe and efficacious, then obviously you get the confidence from that, and you will convey the same confidence to patient as well, or to other healthcare practitioners. Without such evidence in your hand, you cannot convey this. Am I right? Okay. Now, pharmacists are presented with a more number of diseases and possible treatments to consider on a day-to-day basis. So basically, you have so many diseases, so many treatment options. Every day they are increasing. So we are expected to have current knowledge in the fields of expertise and to remain competent throughout their careers. So it is not only that you are studying B Pharm or Pharm D, and after that you stop reading about the drugs. It's not possible, because every time this knowledge is increasing, you are getting more and more new drugs, more and more information about the old drugs which are already existing. So you have to read about them, you have to read more about them, so that you can able to answer to the people. In addition, the number of information sources has ballooned. So we must sort out information from many sources. You can see there are a lot of textbooks, a lot of journals, a lot of the information resources are available currently. In fact, they are increasing again, right? So you cannot say that everything which is published in a journal, everything which comes out of a study is true or is valid, right? There may be a lot of studies which are published without a proper review or proper peer review. Probably there is a possibility of having this false information exist in many places or in many journals. So you have to have evaluation procedure. You have to do your own evaluation, right? For example, you are purchasing something, or maybe some electronic instrument. What you do, you read about it, am I right? You read about it, you understand about it, and you try to ask people in and around you about this particular electronic gadget. Then if you are satisfied, you will go for that. Similarly, to believe certain studies or to believe certain information, you have to evaluate it. How you can evaluate it? Maybe you attend some seminars, from that you get knowledge. You read journals and then try to evaluate it. You discuss all these things with your colleagues, maybe your other people working with you. So they also give you some idea. And then you also look into guidelines which are released by organizations like WHO, ICMR, DCGI, FDA, and so many other organizations. So these organizations give you guidelines. So you try to believe them. Government agencies and expert committees, organizations, all this you will look into before believe something, right? Now, to believe that the Russian vaccine really works for the COVID-19, you have to have some guidelines available from WHO or ICMR or FDA or DCGI. Somebody have to give you some guideline to believe its safety and efficacy, right? Otherwise, you will not believe, you will not recommend that to your patients, am I right? Or you will not share that knowledge with any patient. So basically, to believe any one of these research which is available, you have to have some organization to back up that. Or you need to have your own evaluation criteria. You have to spend time on evaluation of that clinical studies. Okay, so basically evaluation of clinical studies is a part of an overall approach of evidence-based pharmacy. Now, in the evidence-based pharmacy, the main criteria of the evidence-based pharmacy is evaluation of the studies. So before believing anything, you have to have an evidence for that. And based on that evidence, you have to practice your pharmacy, or you have to practice your science, you have to practice your clinical science. Basically, whatever evidence you have to evaluate it, right? So the first step is you have to ask for an evidence, you have to look for an evidence for anything to believe, first of all, and then you have to evaluate it. Without an evidence, don't believe anything. Without evaluation of that evidence, don't believe that, right? So you have to have an evidence, and that too, it should be evaluated by you. Then you can believe that evidence as a strong evidence to believe certain things, to believe that science, and then you can believe it, or you can follow that, or you can recommend that to your patients. Okay, so what is the definition of this? EBP, or evidence-based pharmacy, is applying the best evidence that can be found in the literature to patient with a medical problem, resulting in the best possible care for each patient. It's nothing but looking into the literature for the best evidence available for the treatment of that particular patient. So you need to have the details about the patient, you need to have the details about the case, and you need to put it in a particular format. It is out of the scope of this particular lecture, but that is also very important, putting it into a particular format. So after putting it into a particular format, you will understand the case thoroughly, and then you will go into the searching of the literature. Once you search the literature, then you can find a lot of evidences, and one of the evidences, some of the evidences are true, and those true evidences are those good evidences you are going to extract from that literature, which you are going to use it. And this format of writing a case for searching in the literature is called PICOT format. It's called PICOT format, P-I-C-O-T, right? So you can search about this in the Google for more information about PICOT format. Please learn about it. It's very, very important. If you already not know, if you already know it, it's fine. Otherwise, please search for it. Learn about it. It's called PICOT format. Please remember or note down PICOT, right? PICOT is PICOT format. Anyway. Okay, so once you write your case in the PICOT format, you are now ready to search for the information. Now, there is another definition for evidence-based pharmacy. Evidence-based pharmacy is an approach to healthcare practice that uses the results of patient care research and other available objective evidence as a component of clinical decision making, right? Basically, to have this evidence, and to evaluate the evidence, you have to follow certain procedures, and those procedures we are going to look at. Okay. Now, as I already told you, you have to have an evidence to believe anything, right? If somebody say, okay, my vaccine works best, my vaccine is well studied, okay, that's fine. I agree with you, but I need an evidence to believe it. I need a strong evidence to believe it. I need all the results of whatever studies you have done in whatever number of patients may be. I need all that details so that I can believe it. if this comes out, if this literature comes out, or if this results comes out, then I can study about that. You can study about this, and then you can believe or not believe. That's the result of that particular study. Okay. Okay. In evidence-based pharmacy practice, a number of skills are required. For example, the first skill is the ability to define a criteria such as effectiveness, safety, and acceptability. What is the criteria? The ability to find the evidence. You have to find an evidence. The ability to assess the quality of the evidence. This is what we are going to discuss today. The evaluation of clinical research comes under assessment of the quality of evidence. If you are able to assess the quality of evidence, that means you have a technique or you have the knowledge to omit whatever the studies which are not following proper guidelines, which are not following proper ethics in conducting the studies. You can able to, or you are able to omit them. You are able to filter them. So once you filter all those bad studies, you will get you will have the remaining studies will be believable studies, or the studies which you can depend on, and then you can proceed with the studies. The ability to assess whether the results of the research are applicable to the whole population from which the subjects have been drawn, right? So then you need to look into the sample which is used for the clinical research, and if they have used the proper sampling techniques are used, then you will understand whether I can utilize this knowledge for the or the clinical research results for the entire population, or it is only restricted to a particular group of people. The ability to assess whether the results of the research are applicable to the local situation or not, right? To your situation, to the current situation or not, you can understand from the evidence-based pharmacy practice. So to continue with evidence-based practice, you have to have all the skills. Okay. So one of the important skills, as I told you, is to evaluation of clinical research. That is nothing but assessing the quality of evidences. Assessing the quality of evidences. Now, once you have this assessment knowledge or an ability, probably you can practice evidence-based pharmacy successfully. You can practice EBP successfully, right? Otherwise, you will believe whatever is available in Google or whatever is written in the media, and you will believe all of them, right? And then you will end up in giving some wrong therapy or suggesting a wrong therapy to your patient, right? So as I told you, why pharmacists must practice evidence-based pharmacy? Because he is not only involved directly in the healthcare, he is involved in research, he is involved in clinics, he is involved in community. So your services are available everywhere, right? So your available everywhere, your available or your services are required everywhere. So if you do not practice this evidence-based pharmacy, you will end up in giving them some suggestions which are not correct, which are not accurate, right? So that is, need to be avoided. So you can avoid that situation by practicing this evidence-based pharmacy. Now, from where we get this clinical research, from where we get this information about the clinical research, about the various studies, you will have some primary literature available in the form of original research results published in the journals, dissertations, conference proceedings, correspondence with various scientists. They share results with you or to your organization. So this is the primary literature. There are many important journals, right? NEJM, JAMA, right? Or many other journals like Nature, for example. Secondary literature is also available, like abstracting and indexing services, review articles, systematic reviews, meta-analysis, practice guidelines, which is available from many sources like PubMed, CINAHL, Cochrane Library, Web of Science. One thing I must tell you, these are directed towards different groups of people, right? For example, CINAHL. CINAHL database is directed to give the answers about nursing, right? Or the nurse care or nursing care, right? Cochrane Library publishes review articles. So each one of them is directed towards a particular group of people. So secondary literature, primary literature, you have to have a knowledge where you have to go, which part of the literature or which part of the agency or a website give you the exact information. You have to have an idea about that, right? Then you can reach there to that particular agency. Tertiary literature is also available, like textbooks, encyclopedias, handbooks, some of the newspapers which publishes health columns and other things, health related information. So some examples are also given here. Goodman Gilman's book on therapeutics. So you have primary, secondary, tertiary literatures. From these literature sources, you get the results of the studies which are published or which are available. Then you need to evaluate them. Okay. Now, important parameters for evaluation of studies. Now, before going to the important parameters for evaluation of studies, I must tell you the literature sources itself is a very big subject. Now, you all know that we don't believe all those literature sources which are published. If you see the journals alone, there are thousands and thousands of journals in India itself. And if you go out of India, you will find so many thousands of journals published about medicine, about the medical sciences, about healthcare sciences, about pharmaceutical sciences, and so on. Now, all these journals do not maintain the same quality standards of peer review, right? You know what is peer review? It's nothing but expert review. And they don't maintain the same quality standards in peer review. So you believe certain journals and you don't believe them. That is a very important point. You have to have a knowledge about the number of journals you have and how many of them you can believe, how many of them you cannot believe or you need to ignore, right? So those things you have to have a knowledge. It itself, the literature sources itself is a very big topic. You please look into the literature sources more deep to reach to the literature very fast, to reach to your required answer very fast. How to use PubMed, it itself is a very big science, right? Basically, all these indexing databases are developed based on algorithms, certain computer algorithms, right? I'm not expert in that a particular concept or particular science, but you can read more about it. Actually, that's a wonderful science. The computer algorithms, based on that, these indexing agencies like PubMed work based on that. So some time back, I told you that you need to have a question. You need to have the literature survey question in a certain format called PICOT, P-I-C-O-T, right? Why we have to have this in the PICOT format? Why there is a need for formatting the question, right? Why can't we ask like, okay, I need information about vaccine, so I will type COVID vaccine 19 in PubMed, right? So what is the result of that? You just try yourself, COVID vaccine 19. You just type that in Google Scholar or PubMed. You will get thousands and thousands of pages of information. Now, the problem is you don't have that time, you don't have that energy time to read all these thousands and thousands of pages of information and identify or reach to a particular answer, a perfect answer for your question. Got it? Then you need to modify your question. You need to modify your question into PICOT format so that the number of answers will be reduced. The number of pages will be reduced. The less number of pages, more accuracy to reach the particular answer, so that in short period of the time, you can actually reach to the correct answer. There is a more chance to get the correct answer with this particular format called PICOT format, because all these databases, what we are saying, what we are discussing now, like PubMed or Science Direct, they developed using certain computer algorithms like Google Scholar. They understand the question in certain format, right? So in PICOT format, you have to give details about the patient, you have to give details about the treatment, you have to give the details about the outcome of the treatment, you have to give the details about the time frame. That is nothing but PICOT. Now, if you put your question in this particular format, the 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 answers you get will be more faster. You'll get more answers more faster. You can reach up to the answer more faster. Whatever PubMed gives you, or whatever answers the PubMed gives you, will be more suitable to your question. So there is a more possibility of getting the correct answers. So please try to read more about this, right? So that itself is a very important subject. So if you are interested, please read more about the PICOT. It's not only for the people practicing in a hospital or a community pharmacy, but it is also useful for everybody, right? It is useful for everyone, right? Like tomorrow, if you are into research, you need to search for literature, am I right? And when you are searching the literature, you have to have knowledge how to search it, right? And how to search it in PICOT format. Okay. So what are the various databases you have? You have to have a knowledge about it. Each database is targeted towards a particular group of population. You have to have a knowledge about that. Then your search is authentic, your search is valid, your search is accurate, and your search will lead to certain answers. And those answers will be more valid for you, for your question, and you can believe such answers after evaluation of that. Okay. Now comes back to the parameters for evaluation of the studies. Now, while evaluating a study which is already published or reported, which is available with you in your hand, the first and foremost thing you have to look into is the study design. What type of study design they used? What are the study objectives and hypotheses they have? What is the sampling, randomization, blinding techniques they used? What are the measurements they used in the particular study? Whether this particular study is peer reviewed, expert reviewed or not. Conflict of interest. Is there any conflict of interest? So these are 6 very important points you have to look into. So now for a practice, you can also do one thing. You can find some study which is done recently and download that paper which is published, and then look into its study design. Write it down, what type of study design they used. What are the study objectives and hypotheses they had? What is the sampling techniques, randomization, blindings they had? All this, you please note it down in a paper. And then you start evaluating each one of them based on the parameters which we are going to discuss now, right? Based on the parameters which we are going to discuss now. Now, after that evaluation, you will understand whether you need to believe this particular information or not. If you cannot believe it, or if you understand that this particular study was not done properly, then obviously you have to look for some other study. Okay. Now, the first one, types of studies. Now, the study design many times is either a descriptive study or an observational study or an experimental study. These are basically 3 classes or categories of studies we have. Now, the descriptive study is simply recording information from observing the patients. You just observe it, and then they do the observational study, and they will identify that whatever results they found, and they will write it down in the form of a case report or a case series, and then they publish it. Okay, these are descriptive studies. Then there are observational studies. You have case control, cohort, and cross-sectional studies, right? And then there are experimental studies like controlled experiment, non-controlled experiments. Actually, the basic difference between descriptive, observational, and experimental studies is, in case of descriptive and observational, there is no intervention. But in case of experimental studies, the investigator actually intervenes in the study, right? So you need to find whatever literature which you got is the literature followed the descriptive pattern, followed observational pattern, or an experimental study pattern. You need to identify that. Many times they express that directly in their methodology section. Sometimes they will explain how they have done this study in a detailed manner, right? That depends on each type of study, right? So some studies, they just give, right, how they did in just 1 or 2 lines, they will explain about it. In some of the published literature, published studies, you can see they describe about this study pattern in a detailed manner, right? So which is very, very useful for you to understand what type of study design they used. Controlled experimental designs are best since they are use a treatment group and a control group, right? So in the case of experimental studies also, if they have controlled experimental designs, that will be considered as the best, right? But can we do controlled experimental studies in all the clinical research which is done? Definitely no, right? It's not possible to conduct experimental studies, that too controlled experimental studies in all type of experiments. But in some studies it is required, in some studies it is possible and required. So you need to search for that. So why control group? Why we need to believe this most? Why we consider this as, you know, 5-star study, or why we consider this as a gold standard study, right? Why we need to have this control group? What is the reason for having this control group? Excuse me. Yeah. So the control group is necessary to uh, account the factors that might affect the study results. So you need to have the control group to identify those factors, to nullify those factors which are affecting the study results. Investigators compare the effects seen in the control patients with those in the treatment patients to determine if there is a difference between them. You have different types of controls like placebo, active, no treatment control, or a historical control. Now you need to understand what type of control they used in the clinical research. First of all, what is the type of study design they had? Is it descriptive, observational, or the experimental study? If it is experimental, is it a controlled experimental design or not? You need to identify. If it is a controlled experiment, what type of control they used? Is it a placebo control or an active control? A no treatment control or a historical control, right? So basically controlled experimental designs are considered as the best, right? You need to keep this in mind. Okay, there is a one more study design you will observe sometimes. It is called NF1 study design. One unique type of experimental study has been called the NF1, or a single subject research design. Here the patient is just one person. The researcher conducts a baseline assessment of the patient's condition, followed by therapy initiation. So, a researcher convinces the patient to participate in a study, then understand the baseline assessment, do the baseline assessment, baseline parameters he will observe, and then he will initiate the study. It can be beneficial for studying individuals with rare conditions or those requiring unique individualized therapy. So when you have a patient with a rare condition where you don't have, you know, chance to find more number of patients with a similar disease, then obviously you have to adjust with this NF1 study design. Now, what are the disadvantages of this particular study design? Inability to generalize results to others. You cannot extrapolate these results to any other group of people in the society. Difficult to impossible to perform statistical analysis. It is not possible. Difficult to validate the findings. So basically there are disadvantages, but this type of study design also exists, right? So you need to read more about the methodology, to read more about the type of study design they used in order to understand whether we can extrapolate these results to our population or not. If this kind of study design is followed, if this study design was adopted by someone sitting in Africa or America, and they completed a study and published the results, and with the published results are, the promising, based on that study design, you cannot start treating your patients in India, because the number of patients they used in that particular study was just 1%, right? So you cannot just believe that, right? You cannot just follow that, right? Maybe you need to wait some more time to get this kind of study conducted on more number of people. Particularly, the more number of people should have the Indian population as well, right? Because of the genetic differences we have between the different populations of the world, right? So you need to wait until you get some study which is believable in nature, right? But this kind of study design is also exist, right? You can find them in the clinical research. But the value for this study design is very less, because they used only one person. Now, if you look into observational studies, again, the value for this observational studies are also very less, right? When it comes to evaluation of clinical research, you don't consider them on par with the experimental studies. Now, what are the examples of observational studies we have? Case control, cohort, cross-sectional studies. These are observational study designs. The treatment taken or other exposures studied were not given by the study investigators. Basically, the difference between observational and experimental is, as an investigator, the person who is investigating is not having any say in the treatment. He is not going to, suggest any treatment. Observational designs are generally used when it is not possible to conduct controlled studies. But whenever there is a possibility, you expect the experimental study results. You expect to have the experimental study results. If there is no possibility, of course, observational study results are now evaluated, and they will be believed, right? They will be the best observational studies you will look into if experimental studies are not available. If experimental studies are available, obviously you don't believe or you don't look into observational studies, am I right? So then why people do observational studies and publish them? Why people do case control, cohort, and cross-sectional studies and publish them? Because they publish observational results, because observational studies can become a basis for the future experimental studies. Why the people do observational studies? Because observational studies can become the basis or a hypothesis, right? To generate a hypothesis, observational studies are useful. Now, from the observational studies, you have an hypothesis with you, and based on this observational study hypothesis, you will consider doing or taking up the experimental studies. You will do experimental studies based on the observational study results, okay? For that reason, observational studies are important. But to treat a patient, to believe the results, it is better to look for an experimental study if they are available, right? Wait for them, right? So wait for the experimental study results to believe and start treating your patient, right? Or start suggesting someone to treat a patient. Now, as I told you, it is this concept of evaluation of research is not only for the hospital pharmacist or a community pharmacist, it is also for the research pharmacist or the manufacturing pharmacist or anybody, right? Basically, because whatever you do on a day-to-day, you have to look into literature, right? You have to read the literature, understand the literature, and then decide whether to believe that literature or not. Now, this believing or not believing the literature is simply based on the evaluation criteria, what we are discussing now, right? Observational designs are generally used when it is not possible to conduct controlled studies. As I told you, in rare conditions, there is no chance to have the experimental studies. So you need to have the observational studies, and this observational studies you can believe or you can start following them when you are not having any experimental studies in your hand. Now let's see one example for an observational study. coffee consumption causes pancreatic cancer is the aim of a study, right? So you have aim in your mind that I want to see whether coffee consumption causes a pancreatic cancer or not. Now, how you do this experimental study? If you want to do an experimental study, what you have to do? You have to recruit patients. Yeah, sorry, you have to recruit the people, and then ask them to take coffee every day. In different groups of people will take different amounts of coffee, right? So low, medium, high, right? You can say three groups, for example. Low group take less coffee, maybe 2-3 cups a day, and then medium group will take 6-7, and the high group will take more than 10-15 cups of coffee every day, right? So you divide them into 3-4 groups, and then you will wait for a few days or few years, so that one of the groups or some of the people develops pancreatic cancer, and then you write your report saying that coffee causes pancreatic cancer or not, right? Whatever may be, whatever results may be. Now the main problem here to conduct this kind of a experimental study is ethical, am I right? Now we cannot conduct this kind of a study as an experimental study. So basically this is not possible, because ethically this is not acceptable to conduct this kind of an experimental study. But the same aim you can use for conducting a study as an observational study. So unethical, so you cannot proceed. Now it is unethical because the investigators administering a therapy to many subjects, to the study subjects. So that means you are telling the people who are not actually taking any coffee currently to take more number of cups of coffee. That is really, dangerous for his health. So you cannot go with this particular concept ethically. Cancer takes a long time to develop, so an experimental study will also not practical. So based on the time requirement, again, experimental study is not possible here. So what is possible now is observational study. An observational study design enrolling subjects already drinking coffee or diagnosed with pancreatic cancer is appropriate here. Either you recruit people who are already having a habit of taking high amount of coffee every day, right? You recruit those people, and then you observe their lifestyle, and you observe them for many years until they develop cancer or some disease, and then you write your report. Or you identify those people who are already having pancreatic cancer and ask them a question: how many cups of coffee they drink every day, or having a habit of taking coffee every day, right? So based on that, you can link both of them. You can try to link both of them, right? This is also possible. So either taking patients who are having a pancreatic cancer already, or people who are having a habit of taking coffee already, right? You're not suggesting them to take coffee. They already have a habit of taking more cups of coffee. So you observe their life, you observe them for certain period of time, and then decide whether coffee really causes pancreatic cancer or not. This is nothing but an observational study. So observational study is possible if you have certain aim like this, but experimental study is not possible, unethical, and it takes more time as well. So it is ruled out. So if somebody did observational study for this particular aim or this kind of an aim, you can still believe those observational studies, because there is no chance to get the experimental study results of this particular study. There's no chance even in the future. So you have to believe these observational studies. Based on these observational studies, you can tell to your patient that don't drink more amount of coffee, right? So this is basically an idea to understand what type of study designs are possible in the observational study, what type of study designs are possible with experimental study, right? So that also you have to look into. Okay, so if you look into the different types of observational studies, I will not discuss more about them. You can read more about these types of study designs. Each one of them have its own, the study pattern, right? Now, why we have to know about this? I will tell you. If A clinical research report says they used a cross-sectional study, right? And cross-sectional study belongs to an observational study pattern. And cross-sectional study have a certain methodology to proceed, right? Because cross-sectional study happens in the present time, right? So they have certain people with an exposure of a particular drug or a particular toxin or something else, and it leads to a particular side effect, or it is going to remove a particular element or reduce the particular symptoms, right? So you have an exposure and you have an effect of that exposure on a disease, right? So this is entire thing is happening in the present times, right? So you need to check whether they exactly followed that procedure or not while evaluating a clinical study. You know what is cross-sectional study, you know what is case control study, you know what is cohort study, and then now you check whether they followed this exactly or not, whether they deviated from the study design or not. If they have not deviated, that means they have followed it properly, right? So that means you can give them maybe a tick mark, right, in the first evaluation criteria, right? You can tell them, okay, your study design is right because you have followed whatever you have written. You have written cross-sectional study and you followed it thoroughly as per the study design, as per the requirement. You have done that. That is why knowing about the different study designs is very, very important to evaluate the clinical research. Then you have a case control, then you have prospective cohort study, right? So these are some of the study designs you have, and which helps you to basically understand whether the study design they followed, whatever clinical research you are evaluating, is up to the mark or not. Now, which one is best? Due to their possible disadvantages, observational studies cannot prove that a drug or an exposure caused a certain outcome. So basically, observational studies cannot prove that a drug or an exposure caused a certain outcome because there are many, many factors which are affecting the disease. Only a well-designed controlled experimental studies can prove that this particular drug is causing this particular effect. Observational studies can still provide very useful information. As I told you, observational studies can become a good basis or a good hypothesis for conducting an experimental study. Until unless you have this hypothesis or a strong basis, of course, you will not spend more time. People will not spend their time, energy, money on experimental study. So they need to have a hypothesis, a strong hypothesis. From where they get this strong hypothesis? From the observational studies. So if they have the observational study backup, if they have a strong observational study with them, obviously they will start working on the experimental study, and they will have a strong hypothesis to conduct an experimental study. For that reason, you need observational study. And as I already informed you, observational studies are required to conduct in certain with certain aims and objectives, like as I told you, caffeine and cancer, right? So where it is not possible to perform an experimental study, in that case also observational studies are done. Observational studies are very important at that time. Now, based on the levels of evidence we have, based on the levels of evidence, you can divide, you can list these study designs in an order of importance and strength of the evidence. If a clinical study is done and if it is thoroughly reviewed and based on this clinical studies, based on the clinical research, some renowned scientist wrote a systematic review or a meta-analysis, and this particular systematic review was peer reviewed, expert reviewed, and then it is published in a particular authenticated journal or a particular journal which is very considered authentic journal. Then you can believe those systemic reviews or meta-analysis. So that will be the number one evidence for you, right? That will be the number one in your order, right? When you are saying, when you're listing the order of the importance, this will be the number one. And the second one is the randomizer control trials, which are conducted thoroughly, and then which is peer reviewed, expert reviewed, and then published, right? After that, please don't believe, even after publication, please evaluate it yourself, right? So please look into study design, randomization, blinding, and various other parameters which are listed, right? As I told you, there are 6 parameters, and those 6 parameters, please look into. Even after publication in a very good journal like Lancet, for example, right? Don't believe them blindly, right? Download it, read that particular study, look into its study design, look into randomization, look into blinding, look into other parameters, look into conflict of interest as well. Then you decide whether to believe that study which is published or not. So then randomizer control trials have the next important one. Then the next important one is cohort studies, then the case control studies, then the case series and case reports, which are basically descriptive studies, as I told you, having very less importance. Then you have editorials and expert opinions and letters to editors and all that, which comes under the 6th one, which is having a least importance to believe, right? So if you want to believe certain things, if you want to believe a clinical research, look for a systematic review written by a renowned scientist, published in a peer-reviewed, authenticated, well-established journals. That will be the number one priority for you. If that is not available, search for randomizer control trials, again peer-reviewed, expert-reviewed, published in a good journal, and then evaluate yourself. Then cohort studies, then case control studies, in this order, please go, right? So then you have now an idea what to believe, what not. So after having this lecture, probably your way of looking into the literature will be changed, right? You will start looking into the literature more keenly, maybe more with more interest into the study designs, not only the results. Many times what happens, we look into the results. The first thing we do is we open the results and we read the results and the conclusion and say, okay, this is published in Lancet, this is published in Nature. So I believe this result and I believe this conclusion, and we follow that, right? But before going to the results, please look into the methodology section. How to apply this knowledge to practice? Now you have this knowledge. Now you know what is the study design, importance of this study design. If a news report claims a drug causes an adverse outcome from an observational study, this might not be accurate. If a news report says a drug causes an adverse outcome, please don't believe it. Please don't believe that news report. You please search for randomizer control trials. You please search for some systematic review or a meta-analysis done, conducted, published in a peer-reviewed journal, and then decide whether to believe that information or not. Further investigation is generally needed for confirmation. You need to investigate, you need to check, you need to evaluate then the literature which is available. For the weakest study designs like case control, cross-sectional, retrospective cohort, confirmation of the results by further study is even more important. So you have some published data based on case control. Don't believe it. Wait until you get the experimental study, right? If there is a possibility. If you know that there's no possibility to have the case experimental study, then obviously you have to depend on observational study, but that is an exception, right? But in general, you have to wait for experimental study. You have to search for experimental study results in general. There are rare cases where only observational studies are possible. So only that is available. So you need to depend on that. Now, in the controlled experimental designs also, there are differences, right? So there are basically 3 types of controlled experimental designs: concurrent control, crossover design, and time series design, right? So these 3 will be explained to you or will be present in the methodology section, written in the methodology section. Now they will write whether they followed concurrent control or a crossover or a time series design. For example, you got a paper or a published literature where all this explanation is not given. They have not given any information, for example, regarding type of study design they used. Can you believe that? I don't think so, right? If I'm in your place, I will not believe that, because they have not given any information about the type of study design they used. How I can believe it, right? So they have to give all this information, how they followed this, how they conducted controlled experimental design. Now, when I am evaluating the study, I will check, is it a controlled experimental design or not? Then I will put a tick mark. Then the next thing is it a concurrent control or a crossover design or a time series design? If it is concurrent control, it is having its own advantages and disadvantages. If it is crossover design, it is having its own advantages and disadvantages. If it is time series design, it is having its own advantages and disadvantages. So I will look into those advantages and disadvantages in that particular study, right? I will try to understand in depth whatever the methodology they followed. So this is the basic idea of these three types of study designs: concurrent control, crossover, and time series. Now you can understand. So in concurrent control, you have a study sample, maybe 100 people, for example. 50 of them will become control group, another 50 will be the treatment group. In crossover design, again, you will divide them into control and treatment, but those who participated as a control group, the same sample will become a treatment group after a certain period of time, right? Of course, you have a washout period in between. And this is the basic disadvantage of this particular study design, because if you are not maintaining that washout period sufficient time, then there is a possibility that some of the patients who participated in control, when they participate again after some time as a treatment group, will carry those symptoms or carry those side effects of this control group. So there is a possibility of the disadvantage, right? It's called lack of washout period or less washout period. There is a time series design also. Here also there is a control group, and the entire population will act as a control group, will be tested as a control group, will be participating as a control group. Then the same group after washout period will become treatment group, right? This is called time series design. Now, as I told you, each one of them have its own advantages, disadvantages, right? So concurrent control, are patients comparable? Because there are two groups of different patients. Now, unless you balance these two groups with a proper randomization, these two groups are not comparable. Am I right? You have to have a proper, strong randomization. Now, there are many techniques of randomization. Please read more about randomization as well. There are many types of randomization: simple randomization, stratified randomization, and so on. Please read more about it. Not all these randomizations or convenience randomization is also there. It's equivalent to no randomization. So all these randomizations are not equally effective. So you have to look into the types of randomization they used in order to believe those study results. Now, particularly in concurrent control, randomization is very important, because you have two groups of different people, and in group A there are certain number of people. In group B there are certain number of people, and you have to have balance among these two, and they need to be similar. These two groups should be similar, not same, but similar, right? So when you want to have the similar groups, and when you want to have this two similar groups for conducting a study, you have to have a randomization. You need to have a clear eligibility criteria. Random assignment is very important to balance these groups. Please remember, random assignment, if it is not done, then CC design, concurrent control design, is going to be a disadvantage for a controlled experimental study design. In the case of crossover design, also there are certain disadvantages. It requires more time to complete. Washout period is very important. And this washout period is depending on the type of drug you are using, right? So you know that certain drugs stay in the body for a long period of the time. And until this drug goes out of the body, you have to have a washout period, right? So this washout period is very important. Then time series design, carryover or other sequencing effects can be seen in the time series design, right? So you have concurrent control, you have a problem with the randomization here, you have a CO design, you have a problem with more time as well as washout period. TS design, that is time series design, carryover or other sequencing effects is the problem here. So you have a disadvantages in each one of these study designs. You have to look into these study designs and understand or check what methods they followed to overcome this particular disadvantages. For example, you have a controlled experimental design where they used concurrent control. You need to look into random assignment. What type of random assignment they used in order to overcome the disadvantages of CC design? You have to have this understanding. From where you get this understanding? Again, by looking into the methodology section carefully, by looking into by doing evaluation of the clinical study properly, you will understand this, right? The best out of experimental designs, the type of control selected, whether placebo or active, should be appropriate for the purpose of the study. Whatever type of control they used, it should be appropriate. The concurrent control design is preferred since its advantages generally outweigh the disadvantages. So concurrent control is generally preferred. The time series design is least desirable, is considered least desirable. Unlike a crossover design, effects of time and drug sequence and the study findings cannot be determined. Why time series design is least desirable? Because its effects of time and drug sequence and the study findings cannot be determined. So what is the best out of these three? Concurrent control. Now, if somebody did an experimental study, that is good, used the control, that's wonderful, and then used the concurrent control design, it's very good study, it's very good advantage it is having, right? So that means it is passed in your first test, right? It is passed in your first test. And if they use correct randomization procedure like stratified randomization, if they used blinding as well, triple blinding or a double blinding, then obviously the study is conducted correctly, wonderfully, and then you can believe those results and you can extrapolate these results to your patients. Now let's see an example. Investigators conducted a randomized single-blind placebo-controlled study of galantamine and rivastigmine given for 12 weeks in 60 patients aged 65 to 85 years with a mild Alzheimer's disease. The investigators state that this study is being conducted to determine the efficacy of galantamine and rivastigmine on quality of life in Alzheimer's disease patients. Okay, this is a basic idea. This is conducted as a randomized single-blind placebo-controlled study of galantamine and rivastigmine. How would this study be conducted using a concurrent control study design? How the same thing can be done as a crossover design? Can we conduct? Definitely yes. We can use any one of these study designs. Then how to make this, right? Now, why this exercise is very important for you? To have, to remember what type of study designs we have and how they are useful for us to conduct a study, and which one have a more value for you, right? Which one is more valuable to us? Which type of study design is more valuable to us? For that reason, this kind of an exercises are very important. Now let's try to answer this. In case of, if you want to do the same study in the concurrent control design mode, randomly assigned to receive either G or R for 12 weeks, they would only receive this one therapy. So in concurrent control, as you know, so the G or R is taken. So they would only receive this one therapy, not two. In the crossover design, the patients would randomly assign to receive either galantamine or rivastigmine for first 12 weeks. A washout period you will maintain. And after that, after 12 weeks, after the washout period, you will exchange the groups. The galantamine and rivastigmine is again given to different groups. And for again 12 weeks, you will observe the efficacy of the drug. So the same study can be done using different study designs. Even though it is an experimental study, you need to look into the type of actual study design they used, whether they used concurrent control design, crossover design, time series design, and so on. Since patients receive each of the therapies in a crossover design, notice that total study duration is longer, obviously, because you are waiting for the completion of one study and then you will wait for washout period, and after completion of washout period, again the same group will receive another drug. Why we have to look into all this? For validity, measure what is desired, whether we are able to measuring what is desired, what should be measured or not, whether that particular clinical research is measuring what need to be measured or not, then whether that particular clinical study is reliable, reproducible and consistent or not. If it is not reliable, if it is not reproducible, if it is not consistent, then obviously how can you believe that? How can you believe and use that particular information for the benefit of your patient? Then sensitivity, is it sensitive or not? Can identify or detect the presence of condition. Specificity, is it specific to the current condition, characteristic, current population, your society or not, right? So if it is not specific, again, you cannot use it. So to know all this, you have to evaluate your clinical studies. Now, finally, study must be appropriately designed in order to fulfill its hypothesis and objectives. So a design of study is very important to believe that results. If the study is not designed properly and if it is still published, still available in the literature, please don't believe it until unless the study design is satisfactorily conducted. And the method section specifies precisely how the study was conducted. Serious weaknesses in a study can invalidate its findings. Serious lapses are possible. The use of controls, randomization, blinding are helpful to analyze and draw valid conclusions from a clinical studies. You check what is the control they used, if at all they use it something, and then check randomization, check blinding and all those parameters. And of course, the conflict of interest, those 6 parameters which are given in the one of these earlier slides. Please look into those parameters. Check whether your clinical studies, whether your literature agrees with all of them, satisfies all those parameters, then obviously you can believe it. Otherwise, please don't believe those clinical studies. So these are some of the references, wonderful books written on evidence-based pharmacy. You can read more about them. There are other literatures as well available. So please read more about them, about evaluation of clinical studies, about the concept of evidence-based pharmacy, which is really interesting, and then try to understand PICOT format, right? PICOT format, PICOT, PICOT format, to frame your question, right? So once you are done the framing of a question, then the computer algorithms can understand it very effectively. One more important thing I should tell you, PICOT format is the common format of writing a clinical question everywhere in the world, not only in India, not only in some parts of the world, but everywhere people understand what is PICOT. All the clinicians understands it, the pharmacists understands it, researchers understands it. So the databases which are prepared, the databases which are available also understands that. That is the reason why we need to have a common questioning format called PICOT format, right? So please read more about it. Try to practice how to prepare your questions using PICOT, right? And then use that PICOT next time when you are searching for any literature. So there is a more chance of reaching to the correct literature. Otherwise, what will happen? You typed some question in whatever way you think the best in the PubMed or something, PubMed or Science Direct or Google Scholar, you will get thousands of pages of information, and you cannot read all of them. Obviously, you will take up the first one, whichever comes first will be taken as a answer for your question, and you start believing it, right? That is what happens many times, but don't do that, right? So you try to reduce the number of the answers by using the PICOT, by using other filters as well. In the PubMed, you can see a lot of filters available. You try to understand those filters in depth. There are videos in YouTube. There are in the PubMed itself. There are the literature available in the PubMed itself to read, to find the ways and means to use the PubMed effectively, right? So how to use the PubMed effectively, right? That's also available in the PubMed itself, right? So please try to read more about it, try to understand, then whatever answer you get in the PubMed will be more accurate, more specific, more useful to you, right? To your question. Whatever you do tomorrow, whatever job you have currently or in the future, whether it is in the industry or in the manufacturing or in the research or in the hospital pharmacy or a clinical pharmacy, whatever job you have, you need to have this understanding of searching for a better literature, understanding that literature, and then evaluation of that literature, right? So this is the summary of evaluation of clinical research. Thank you so much. Over to the moderator so that he or she can gather questions and provide to me. Thank you.
Thank you, sir. Thank
Yes,
you very
sir.
much, sir. A lot of questions, and only 2 or 3 questions I will share with you, sir. And
Yeah,
rest of the things I will post for you. And first thing is whether the observation studies can be considered better when compared with experimental studies as a evidential studies, observational studies.
Yeah, that's a good question, sir. Actually, the observational studies can be a good basis, can be a good hypothesis for conducting experimental studies in the future. But to believe those observational studies, you have to have very, very strong reasons. One of the important or very important strong reason is if there is no possibility to conduct experimental study in the future, like the example which I have given in the slides, like coffee consumption and cancer, right? So in that case, there is no chance to get an experimental study in the future. So you have to believe observational study. But in many cases, experimental studies are possible. So wait until the experimental studies are available to believe those results. Otherwise, don't believe observational studies. Observational studies are meant to provide a basis or a hypothesis for future experimental studies. Hope
Yes, yes.
I cleared the doubt.
Yeah, thank you, sir. Thank you. Right. And one more from Dr. Chakka Gopinath. Can NF1 results can be published? The research work can be published or is it possible to publish?
Yes, sir, because there are thousands and thousands of journals we have, right, currently. So there are journals meant for young investigators, there are journals meant for the student studies, there are journals meant for various other categories of people we have, right? So I don't think that is impossible to publish. Anything is publishable nowadays, and there is a possibility of getting everything published, right? So yes, my answer is yes.
And one more thing, sir. Among all these designs, which one is the best one? Whether it is a concurrent control design or crossover design?
Sir, basically experimental study, if it is conducted properly, if they have followed randomization, blinding and other techniques correctly, then if there is no conflict of interest, and after the study, if it is published in an important journal with a proper peer review, then you can say that is a very good study conducted, that results can be believable. But if they have written that it is the study, if they have compromised in any one of these parameters, probably that is the worst study that you should not believe, right? So my answer is, for your question, it should be an experimental study. There should be a controlled experimental study. It should be a randomized controlled experimental study or an RCT, which is actually published in a journal with a good peer review system.
Thank you, sir. Thank you very much. So thank you for your more elaborate explanation regarding importance of literature and its evaluation. the role of pharmacist in evaluation of evidence-based studies, especially concerned to the safety and efficacy, the importance of organizational guidelines, evidence-based pharmacy practice in the treatments, the source of information in SINAR, the parameters for evaluation of studies. So many things you have covered in an elaborate manner. Thank you very much for your informative and most knowledgeable session. Now, I request our principal, sir, to give his final conclude words. Sir. Sir, principal, sir. I think some connection issue is there.
Yeah, it's fine, sir. No problem.
Sir, thank you. So
Yeah.
thank you, sir.
Thank you
Thank
so
you very
much.
much.
Thank you so